A very good morning to Madam Hazwani and my fellow friends. I am Danisha Manikavasin from DBT5P and my metric card number is 23 DBT18F. So today, Silvina Lucas Senior and I are going to present about making transgenic plants by using microorganisms. So first we will start with the introduction of transgenic plants. Transgenic plants refers to the plants whose DNA is modified through genetic engineering technique. This means one or several genes from a different species are introduced and combined with the genetic material of the plant, which changes the traits of the original genome. While horizontal gene transfer has been shown to occur naturally in nature between plants that grow close to each other, various artificial techniques used to insert gene sequence to some plants with the aim of increasing yields, making them more tolerant of various environmental conditions, or making them more resistant to given biotic stresses. The purpose of these transgenic plants is to make it as useful and productive as possible. The examples of transgenic plants are golden rice, flavor sour tomatoes, and BT corn. Moving on to the history, in 1982, the first genetic modified plant was produced using antibiotic resistant tobacco plant, which was resistant towards the herbicides. In 1986, insect tolerance tobacco plant was developed by expressing genes encoding for insecticidal proteins from Bacillus thuringiensis. The first commercially grown genetic modified food in 1994 was the tomatoes, which was the flavor sour tomatoes. This was bred with deactivated gene that kept plant from producing polygalactonase, which is an enzyme that is the starting point for rotting. In 1995, canola with modified oil composition was produced. This is to contain laurate, which is a key material used in the manufacturing of soap, detergent, and personal care products. Before this, only coconut and palm kernel oils contain laurate. In 1999, the first commercial papaya that resistance towards ring spot virus was produced. It has been added with viral capsid protein. This is to create the effect of an immune response. However, the crops were grown in Hawaii. That is all from me. Thank you.
I'm Silvina Dr. Flucosinia. My metric number is 23 DBT 18F 2007. Process. For the first step, we extract the DNA, and the following step was gene cloning. We extract the DNA from the bacteria and we will isolate the extraction DNA. The following steps was gene cloning. We are cloning the gene and we are designing a new gene, which means the extraction DNA was combined into the original gene. And the following step was transformation and tissue culture. We did the transformation for the designing gene, and then we do the tissue culture, which means we grow the plant into the petri dish. The last steps was plant breeding. After the plant was growth, we are breeding the plant. The importance of transgenic plant. The importance was improvement in yield, improvement in insect and disease resistance, improvement in quality, and also its herbicides resistance, and resistant to abiotic stress. And the following one is industrial product, and its rapidly and accurate techniques, and no barrier for any gene transfers. Conclusion. In the future, the research hope to be able to provide the vaccination and medicines in GM foods, which can provide the medical to people in developing the countries. More the transgenic plants offer a new approach to producing and administering the human antibiotic. There are few journals that we use to do the slide and to get more knowledge about our title. Thank you.